Hello and welcome to this course. In last class, we had discussion about the course outline and we studied the nanomaterials and the properties of the nanomaterials. We understand that there are the many factors which affects those properties of nanomaterials as compared to the bulk materials. For example, we understand surface area to the volume ratio. We understand optical properties. We understand these catalytic properties of the nanomaterials. Now, let's understand what is the bionanotechnology and what is the area of research in this topic. So here's bionanotechnology has two terminology that is the if you just understand that there is the bio plus nanotechnology. So it is the combination of the biotechnology and this nanotechnology. So here in this topic, we are going to learn how we can use nanotechnology to understand the biological systems, or maybe we can use the biological system as a toolkit to understand at the nanoscale level. So there are two ways to understand the biological bio nanotechnology. So let's first understand incorporation of nano-scale machine in the biological system. So in this particular approach, we are going to understand how the nano-scale based either machine or maybe the particles can be used to understand the biological system. For example, we have here drug delivery. So we can use biological nanomaterials to deliver that drug to the particular cell or maybe the tissues or organ. And based on that targeted drug delivery, we can increase overall the efficacy of the drug. So we can increase efficacy of drug, and then we can also increase that the and reduce side effect of drug. So there's two advantages. If we use the nano scale machine or the nanomaterials kind of the system, and if we develop the system so that we can deliver that drug to the targeted cell organ, in that case, we will increase overall the efficacy of the drug and reduce the side effect. So these are the advantages. And in the second is the molecular manipulation. So here, using the nanoscale machine, we can manipulate within a cell. For example, if you are looking for the application in the gene engineering or engineering on the gene therapy, so in this case, we can use nanoscale nanorobot or those nanobots that can go inside the cell or in the nucleus, and that change in the gene within the nucleus or DNA. And based on that, we can have the genetic engineering research. Or maybe this kind of this nanobot can be used for the repair of the damaged cell. So here in the manipulations, we can, so this has the application in the genetic engineering field, or maybe we can use this technique for, we can here use these ones to repair to re, to repair damaged cell. So these are the advantages of thus manipulating the at the molecular level using the nanoscale machine. And we have here this third application is the medical imaging. So using nano scale machine, or maybe we can use the nano materials to to improve the overall this imaging applications. We can improve imaging so that we can have this distinct and clear imaging of the biological system. So here the main purpose of doing improving the medical imaging so that we will have this clear imaging of this biological entity, clear imaging of biological sample, right? And and the fourth application is that tissue engineering. So here, using the nanoscale machine, robot boards, or maybe they're using the nanomaterials, we can design the artificial tissues, or maybe we can design that biomimic tissues, so that has this great applications in that particular field. And the fourth point is the diagnostics. So using the nanomaterials based system, we can develop the sensor. So here in diagnostic things, we will either develop biosensors, so that it can use for the early, so that can be used for the early detection of disease. Since nanomaterials have very high surface area to the volume ratio. At the same time, it has more reactive as compared to the bulk material. So that if we use the nanomaterial based system, so then we can diagnose whether that particular person has the disease in the early stage, and then we can save many lives. For example, in the cancer, we can only diagnose the cancer in a patient after second stage. So what about if we diagnose that cancer in the first stage, so we can save the lives of the millions of the peoples in the world, worldwide. So these are the applications of the nanoscale machine in the biological system. Now, the second approach of understanding the bio-nanotechnology is that we can use biological toolkits to construct nano to the microstructure. Here, if you think about, here's this biological toolkit. So here the biological toolkit means that we can use the biological building block, so building block of life. For example, we have here the biological component. We can use DNA, we can use RNA, we can use protein, we can use lipids, we can use polysaccharides. Maybe you can use polysaccharides. So using these biological component building blocks of the biological system, we can develop the materials at nano to this micro scale. So here, using this kind of this materials as a component, as a scaffold, or maybe as a template, and then we can modify this material so that we will have this particular shape and size of that material. So using this kind of the biological component, we will have the desired materials, so that can be used in this, maybe either in the diagnostic application, or maybe we can use in the cancer treatment. And here we have the other here application, that is the assembly based application. So in assembly based application, we can use the three kind of the assembly, and that assembly is either maybe the self assembly, maybe we have this directors direct sorry this is directed assembly, and we have this tem, template assembly. So using three kind of the assembly, we can develop the nanomedicine. So we using that kind of assembly, we have this DNA, we have this nanomaterials. So we can combine together and we'll develop the self-assembly or directed assembly or template-based assembly so that that can be used as a nanomedicine. And this approach has also applications, as I explained earlier, is that this had application like as diagnostics, tissue engineering, medical imaging, manipulations, and the targeted drug delivery. So this approach has the similar application as the first approach. So this is about to understand the bio-nanotechnology using this two way. One is to use the nano-scale based materials. So we can use here is nano-scale based machine and then understand as a biological system. And the second approach is about the use of this biological toolkit and then and construct the materials at the nano and this micro scale, and that has the similar kind of applications, but there's two approach. And if you compare which method is better, so here in this second method is more preferable as compared to the first one, because we are using biological materials, biological component. So here this material, so this approach is more, so this approach is more about this biocompatible, biocompatible. So second approach is the more biological compatible, and it is also the mimics the biological system. So it mimics biological systems. So that's why it has this more efficiency as compared to the first approach where we use the nanomaterials or nano machine to understand the biological entities. Okay, now let's understand what are the field of the bionanotechnology. So in bionanotechnology, we understand what is the biomedical application of nanotechnology, and that is the biomedicine. And we are going to learn the second field is that the self-assembled structure, that is the nano assemblies in the bionanotechnology application. And the third field is the biological research at the nano scale. And the fourth application is about this biomimetics, biotemplating, and the de novo design structure. And our the fourth area of research is the DNA-based nanotechnology and nano-electronics. And the next application is the molecular motors and the devices. Now let's understand this area of research and how you will can contribute if you know the all details about this area, then you can selectively look at that particular area of research, and based on that you can focus maybe on the nano assembly, maybe the nano electronics. So as an engineer, you can look the DNA based nanotechnology or the nano electronics, or you can contribute everywhere. So it depends on your interest. So now let's understand the first area of research of the nanotechnology. Nanotechnology is the biomedical application of nanotechnology. So we are going to use nanotechnology for the biomedical application, and that is the nanomedicines. So in nanomedicine, we have the three main areas. One is the diagnostics. Diagnosis means we diagnose particular kind of diseases by quantifying the biomarker. So that is the term known as the diagnostics. And the second is the implants, for example, medical implants. And here the pharmaceuticals. In pharmaceuticals, you can develop this medicine for this treatment. So these are the area of research in the nanomedicine. So now let's understand the first one is diagnostic. In the diagnostic, if you want to diagnose any patient, we need to have such as one kind of the sensor, and that sensor is called the biosensor because that is used in the biological application. So we need here is the biosensor. So here we need the biosensor. So what biosensor does? So that biosensor has the such kind of the here surface so that interact with our biological sample, or those, or maybe there's here's a inner light. So this may be, so here this may be the inner light, or maybe it is better to write as the biomarker. So these materials may be the inner light or the biomarker. So here we have sensor, and that sensor selectively combines with that biomarker or inner light. And if that combines with the biosensor, that there may be change in the physical change or the maybe the chemical change in this biosensor. And based on that to change either physical or the chemical change, there will be change in the electrical signal. So based on that change in electrical signal, we can quantify how much inner light is present there. So for this one, we will have such kind of we have the concentration, and we have here signal, and we will have the such kind of the plot here, and this is called the calibration plot. So we have the concentration, we have the signal. So this is the any kind of the signal, electrical. So it may be this potential, it may be the current, it may be the impedance change, so any kind of the signals it may be here. So based on this calibration curve, we will know if we get the signal. So from these signals, we will know okay how much concentration of that particular inner light present in mm, that particular patient, and based on that we can know okay this particular patient has this level of the concentration. That means that particular person has that particular disease. So based on that, we can diagnose the patient. Now let's understand here. So here we have the sensor. So we have here sensor, biosensor, and that biosensor reacts with the analyte, and based on the bioreceptor here. So there is bioreceptor here, and that bioreceptors, if that analyte gets with the bioreceptor, so that is change in the signal and the transducers. So you can play with the that transducers, or maybe you can play with the biosensors component. So there are just two ways you can improve the biosensing. First point is the biosensing itself, and second is the transducer. So you can improve the signals by just modifying the transducers or maybe the biosensors part, and then you have the improved signal. So based on these particulars concept, you can design biosensor. Now, if you think about those biosensor, and if you're looking for the in this particular area of research, then we need to know what are the key points for this biosensor. So these are here. They are the main properties of the biosensor. For example, here this biosensor has the capabilities. Here we have the capability to monitor bioanalyte in the real time. So these are the expected from the your biosensor. Second is the it has the enhanced activity, so you can improve the sensitivity of that sensor. And the other one is that it should be the portable, right? So if you so that sensor has the capabilities to monitor in the real time, it has the high sensitivity, and even at the same time it has the uh, portability capability, because if you have to develop very huge, very big biosensor, so in that case you cannot put that you cannot take with you. So if you have the very portable instrument or devices, so that can so person can also buy that one, and that is portable, so that person carry with them. And we have the here other properties like is the high specificity. That means, for example, you want to develop a glucose sensor. So that glucose sensor should be specific to the glucose only. It should not specific, so it cannot react with other kind of the inner light. For example, we have the different foods, so we have the maybe as vitamins, minerals, so that sensor should only specific to this glucose. It should not be sensitive to the vitamins, minerals, right? It cannot be, should not be sensitive to the lipids. So this is about the specificity. Even though if you think about the specificity within the carbohydrate, it should not be sensitive towards the other carbohydrates. For example, starch, it should not sensitive to the sucrose. That should be only the sensitive to the glucose. And at the same time, that sensor should be the low cost. So that should low cost, and it has the self testing and the calibration. So before application of any kind of sensor, we need to do the calibration. So if that sensor has the capability to do the self calibration, so that is very good. And other point is about that minimum response time. MX is the multiplexing that we can use multiple times. Minimal samples treatment we need. That sensor should be accurate, and this wise measurement range, automations, reusability. So these are the key component of any kind of the sensor. We need these kind of the properties of the any sensor should have kind of the reusability, so that we can use again and again. So that will reduce the overall the cost of that sensor. Okay, so now let's understand, for example, based on the example here, if we think about here's the nanomedicine, so that nanomedicine, based on those nanomaterials, we can use that is in the different application. For example, we can use in the radiation therapy, we can use the phototherapy, chemodynamic therapy, and this chemotherapy. So these are the areas of the nanomedicine. So we can develop the nanomaterial based on the medicines, and for that we can use all these kind of the applications. So here we can use the medical imaging, photosensitization, immunotherapies, drug delivery, nano cascades, chemocatalysis, photocatalysis, localized hypothermia. So these are the kind of the application we can develop based on the nanomaterial based system. So these are the application of the nanotechnology. Now let's understand the second kind of application of the bionanotechnology, and that is the self-assembled structures, or maybe those nano assemblies. So here, nano assembly works is that we have this component. Let's understand here we have this component of that nano assemblies. So that components self-assemble, and that particular conditions of the pH or maybe temperature or something. So based on that kind of the self-assembly, so that forms kind of the assembly assemble here. And once it forms the assemble, so that is sensitive to the temperature to the light. So if you put the light here, so in that case, that gives you the particular color. And based on this photoactive nature of that assembly, we can use as a photomedicine. So this kind of the photomedicine has huge application in the cancer treatment and this cancer diagnosis application. So let's understand how this kind of the materials are used in the image guided therapy and combination therapy. So this has a huge application in this cancer treatment and the cancer diagnosis, treatment and the diagnosis. So these are the huge application in these particular two areas. Now let's understand how you can use nano assemblies or self-assembled structures for in the cancer treatment or maybe the diagnosis application. So we can use two kind of approaches, and that are PDT and PTT. So here PDT, so here PDT means that this is the photodynamic therapy. So this is the photodynamic therapy. Or maybe the other name of this PDT is that photo photochemotherapy. So there the other name of the PDT is the either the photochemotherapy. And here the PTT, here's PTT means that is the photothermal therapy. So we can use either PDT or PTT as a tool to diagnose or for the treatment of the cancer. So here in let's understand PTT first. So here PTT, what is that? So here you have component here and that forms here as a self-assembled structures. So in particular case, if you just put the light on this material, so in that case, that material, so here in PTT, your material, so here you have this material, so that material absorb in, so that absorbs near IR radiation, and that will change, that give you the increase, so increase the heat energy, right? So in PTT, use assembly absorbs near IR radiation, and that gives the localized heat energy, that is the thermal energy. And based on that heat energy or thermal energy, that has capabilities to kill the surrounding cells or the organ in that particular thing, cells. But it has a huge application in the cancer treatment. So using this kind of you can kill the cancer cells in body. So here we can use either polymeric assemblies, maybe we can use the semiconducting polymers kind of this organic materials in PTT, or maybe we can use here MOF kind of the materials, nanocomposite materials, or maybe we can use gold nanoparticles, carbon-based materials, or maybe UCNP kind of this materials. So these materials can be used in this PTT application. In PTT, what is that? That is the photodynamic therapy. So in this kind of the materials absorbs the radiations, and that gives you the particular light, and that has capabilities to diagnose the particular cells or organs in the body, or maybe that can kill the surrounding cells. So here, either you use with the PTT, or maybe with PDT, we can monitor the patient's health. And the other third area of the research of bionanotechnology is the biomimetics and the biotemplating. So now let's first understand what is the biomimetic. Biomimetic means, we can say, is the such kind of the biomimic, mimicry. So here we need to develop the materials so that that can mimic the biological sample, that is biological entity. So here we also go, so for example, in biomimicry, if you want to develop the enzyme, so that is the one of the example is the nanoenzyme. So here nanoenzyme is kind of the nanomaterial based enzyme, right? So here this is nanomaterial based enzymes, but in our body that enzyme is the biological entity. So we can develop that artificial enzyme which can mimic the natural enzyme that is present in our body. So here now you ask a question, okay, why we need to design the artificial enzyme? Well, so natural enzyme has low catalytic activity. So if you want to improve the catalytic activity, so for that we can manipulate that site of the activity of that enzyme, and then we can improve overall the activity of that artificial enzyme. And this has a huge application in the cancer treatment, and it can be also used in the diagnostic application. So you can use that artificial enzyme based biosensor, and since you have, if you increase the catalytic activity of that enzyme, then that kind of the sensor has capability to diagnose patient in early stage, because that is more sensitive by the determining the concentration of that biomarker. So these are the application, and the other application is the bio environmental monitoring. So we can use that artificial enzymes to monitor the biology, so environmental sample as well. So these are the area of research in biomimetics. And what about this biotemplating? So here in the biotemplating area of research, we use biological sample as template, and using this biological sample as a template, we modify that kind of this template for our desired application or the desired activity of that material. So here we use biological template. Maybe we can use here as bacteria, or maybe we can use virus, or maybe we can use the DNA, or maybe we can use the lipids. So we can use the biological sample here, and we modify that biological samples bacteria. So suppose if you want to develop the nanomaterial that has a shape of that particular bacteria, or maybe the particular virus. So in that case, we can use that bacteria as a template, and then we can deposit our nanomaterials on that bacteria or the virus. So in second case, we deposit that materials, and then after, in the third step, if we just remove that biological sample, that bacteria from that material, so then again we will have the very good shape of that material of that particular bacteria and this virus, or maybe the virus. So here, if you have here's the maybe the kind of the materials, so we can modify these materials either inside interface or the outside, and if you just remove these materials, then you have this very good growth of that materials and very good desirable shape of that nanomaterials, and based on that we can use for the diverse applications. So these are the area of research in biomedical where we use biological sample as a biotemplate, And the second is the in biomimetics we develop the artificial materials to mimic the natural biological sample. And the next area of research in bionanotechnology is the DNA based nanotechnology and the nanoelectronics. So now let's understand what is the DNA based nanotechnology. So here in the DNA-based nanotechnology, we use as a DNA as a structures material, and then we modify that structures material for the different applications. For example, we can have this DNA here, so we can modify that material with the nanomaterials for the anti-cancer treatment here. We can also modify with that materials with the antibacterial treatment. We can use for the immunotherapy, we can use the gene therapy, we can use targeted therapy, and we will have the promoting cellular uptake of this material. So these are the different applications. So we have a DNA and we modified with our desired nanomaterials and used for the different applications. So these are the area of research if you are just focusing on the DNA-based nanotechnology. And then second is this DNA origami. Since DNA can be modified into the different shape and sizes, so that has the capabilities of use that materials in the different application, therapeutics, imaging, diagnostics, or the different application in these areas. What about the application of the nano-based DNA-based nanoelectronics? So in this particular area of research, for example, DNA-based nanoelectronics, we use DNA as a scaffold and then modify. So if we have here DNA, so here you have here DNA, right? So we can modify that DNA with our desired material. So here you can dope here's the drug, particular the drug. You can modify this end group here, point here with the nanoparticles, and based on this one we can modify that DNA for the diverse application. So here that is the use of that DNA based scaffold, where you can modify that DNA scaffold with the different materials. So here you can modify, so you can use a scaffold and modify it with materials. It is better to write nanomaterial with nanomaterials. Or maybe if we can modify that one with the drug, so that can be selectively used here. Or maybe if you have the here electronics means if you have the DNA and if any particular drug, if you have any particular drugs bind with that DNA, in that case you will get the signal change, and based on that signal change you can use as the sensor application. So these are the so this is the reference. If you are interested, then go there and read in more details about how nano electronics works. And the other thing is that we can use DNA as the molecular wire. So here you have the DNA. So we have the DNA. So this is DNA, and since DNA has capabilities to connect the electricity, so that can be used as a nanowire in this nanoelectronics applications. And again, the DNA has the important advantages in this in the shape and the sizes of that DNA, and that gives the advantage in the development of the nanoelectronics. So these are the important area of research that is based on the DNA. And the next area of the research of bionanotechnology is this molecular motors and the devices. So here we can develop the molecular motor that is based on the proteins. We can based on this DNA kind of this molecular motors, and that kind of the motors can give the desired or specified the work in cellular level. So this kind of the molecular motor can be work at so that can be work at sorry, so this can work at the cellular level. So that will go inside our cell, and that can work there in the terms of maybe it can modify the such kind of the gene in the DNA, or maybe that can repair the cell damage, and that has the huge application of the that molecular motors and devices. So these are the applications. So if you want to read more details about these molecular motors, you go in that reference, and there are other reference here, and study in more details about what is the molecular motors and devices. So in this particular class, we studied how to approach bionanotechnology. So we can approach bionanotechnology by two ways. One is using the nano scale machine to understand the biological system. And the second is that we can use the biological toolkits to develop or construct nano to the microstructures. So using these two approaches, we can have the diverse area of research. Hope you enjoyed this class. So in next class, we are going to learn what is the growth possibility in this in biotechnology areas, what is its key challenges, and what are the key opportunities in biotechnology. So see you in the next class. Thank you very much.